Good morning, children.
Good morning.
We were discussing about different endocrine glands present in human beings. So today also we'll be continuing with the same topic. A few more endocrine glands are remaining. We'll be learning about the hormones produced by them and the functions performed by these hormones. And the next endocrine gland we need to learn is a very crucial gland, and it's having both exocrine and endocrine functions. And as an endocrine gland, this particular gland helps in digestion. Can you identify about which endocrine gland I am speaking about? It plays a very important role in digestion and at the same time produces two important hormones which are very essential. Can anybody guess and tell me which is that? Is
Pancreas.
pancreas. So next we are going to learn about pancreas. Pancreas is having both exocrine and endocrine function. As an endocrine gland, it produces two types of hormones, insulin and glucagon. Both of them play a very important role in maintaining normal glucose level in blood. And as an exocrine gland, it will produce secretions, the pancreatic juice which contain different enzymes. This will be released into the duodenum or the upper part of the small intestine, and it will help in the digestion of food. So that is why we call pancreas as an heterocrine, or we even call it as having dual function, a gland with dual function, both exocrine and endocrine function. So let us learn more about.
Nandana is and the insulin.
Who is waiting?
Nandana.
Okay. So here you can see the picture of pancreas, and the location of pancreas also you can find very close to the stomach. It's a heterocrine gland or a composite gland because it's having both exocrine and endocrine function. So it is a heterocrine or a composite gland because it has got both exocrine and endocrine functions. And there are some specialized cells, some specialized pancreatic cells. You call it as islets of Langerhans. The word meaning of islets is islands, a small island. And it got the name islets because these cells are scattered, or these are irregular patches. They resemble irregular patches like an island. So you call it as islets of Langerhans because these cells were first described by or discovered by Paul Langerhans. I think it was in 1869, and that is why these cells got the name islets of Langerhans. So they form the endocrine part of pancreas, and 1 to 2 million islets of Langerhans are found in a pancreas. You can say 1 to 2%, just 1 to 2% of pancreatic tissue is made up of islets of Langerhans, and they produce two types of cells, alpha cells and beta cells. And these alpha and beta cells will secrete peptide hormones. The peptide hormones are glucagon and insulin. So don't get confused which one is secreting glucagon and which one is releasing insulin. So remember, if you can remember the term BIN, IN, insulin, B for beta. So if you can remember the term BIN, beta cells release insulin. So if beta cells are releasing insulin, then glucagon will be released by alpha cells. They help in maintaining glucose homeostasis, maintaining the balanced normal level of glucose in our blood. So once again I will repeat, pancreas is a composite gland. a heterocrine gland because it's having both exocrine and endocrine functions. About the exocrine function, I already mentioned it produces pancreatic juice which will pass through the pancreatic duct and it will be released into the duodenum where it acts upon food, and this juice is containing different enzymes which will help in breaking down the food particles. And islets of Langerhans are specialized pancreatic cells which constitute 1 to 2% of pancreas, and these cells we call it as islets because they resemble islands, because they are in the form of irregular patches. And islets of Langerhans has got two types of cells, alpha and beta cells. Beta cells will produce the hormone insulin and alpha produces glucagon. Together, these two hormones will together bring about homeostasis, glucose homeostasis. First, we will see the role of glucagon. Glucagon is a hyperglycemic factor. Hyper means this will help in, see, two hormones are there, glucagon and insulin. One will lower the glucose level, the other one increase the glucose levels. So glucagon is a hyperglycemic factor. It will help in increasing the glucose levels. When it goes low, it will help in bringing it back to the normal level. It will increase and bring it to the normal level when the glucose level goes low. Maintain normal glucose level and acts on liver cells, hepatocytes, and stimulates glycogenolysis, resulting in an increased blood sugar. So the blood sugar level can go low from the normal level. So then this hormone will increase its level and bring it back to the normal level. So how it is bringing about normal glucose level? It will act on liver cells. You know that excess glucose. So when you consume food or the excess glucose derived will be converted, because whatever is required by your body will be utilized. Excess amount of glucose will be converted to glycogen and it will be stored in your liver. And what these glucagon will do is, it will act on the liver cells and it will stimulate glycogenolysis, breaking down of glycogen to release glucose. Glycogen will be converted back to glucose by the action of this hormone, and this will increase the blood sugar level hence we call it as hyperglycemic factor or brings about hyperglycemia. It will stimulate gluconeogenesis also, that we discussed in the previous class, formation of glucose from non-carbohydrate sources. When glycogen is broken down to release glucose, it's a carbohydrate source, but at the same time from non-carbohydrate sources also glucose formation will be stimulated by this particular hormone, and it will also reduce the glucose uptake by the cells, the cellular uptake as well as utilization. So the cells will not be allowed to take it and utilize it. So that also will promote a rise in the blood sugar level. So in the blood sugar level will increase because it is preventing cellular uptake and utilization. So that is about glucagon. As I told you, pancreas, the islets of Langerhans, consists of two types of cells, alpha and beta cells. Alpha cells will produce glucagon. You call it as a hyperglycemic factor because it will help in increasing the glucose level in blood. And how do they increase the glucose level and bring it back to the normal state or normal glucose level? That is by two processes. Three processes are there. Two main processes listed are gluconeogenesis and glycogenolysis. And to carry out glycogenolysis, it will act on liver cells where glycogen is stored, and glycogen will break down to glucose that will increase the sugar level in blood. It also promotes the formation of glucose from non-carbohydrate sources. So that also will increase the glucose level in blood. And third method is by preventing the uptake and utilization of glucose by the cells. That also will contribute to the rise in the sugar level of blood. Okay, children? Have you understood?
Yes, ma'am.
Okay.
Yes, ma'am.
Now we are proceeding to the next hormone, that is insulin. So insulin, the other one was hyperglycemic factor. This is hypoglycemic factor. When the glucose level increases in your blood, the role of insulin is to bring it down to the normal level. Glucagon, when the level goes down, it will increase it to the normal level. Functions: regulate glucose homeostasis or helps in maintaining the normal glucose level in blood. Acts on both hepatocytes and adipocytes. Hepatocytes, I told, liver cells, and adipocytes are the cells which the cells of adipose tissue which will store fat. And it is acting on these two cells, and it will promote the uptake and utilization of glucose by the cells, whereas glucagon was preventing the cellular uptake and utilization, whereas this hormone will promote the uptake and utilization of glucose by the cells. So as a result, the blood sugar level will be reduced because it is getting utilized by the liver cells and the fat cells. So the level of glucose present in the blood will be reduced. And such a situation, when the blood sugar level is decreasing, you call the situation as hypoglycemia. It will also stimulate glycogenesis, means formation of glycogen. How glycogen is formed? Glucose is getting converted to glycogen. So when glucose is getting converted to glycogen, again the level of glucose will be reduced in blood. So those are the different methods by means of which the amount of glucose in blood is reduced. So in glucagon, what we learned was glycogenolysis. Glycogenolysis takes place by the action of glucagon. That means glycogen is breaking down to glucose. So the glucose level increases. But glycogenesis means formation of glycogen. So here the glucose will get converted to glycogen. That also will help in reducing the glucose level in blood. So I'll just repeat the points once again. About insulin, you call it as a hypoglycemic factor. The reason is it will help in bringing down the glucose level to the normal. And how it is carried out? It will act on hepatocytes and adipocytes, and it will stimulate the glucose uptake and utilization. As a result, the blood glucose will be absorbed and utilized by these two types of cells, and the glucose level gets back to the normal. And it will also promote glycogenesis. Glycogenesis means glycogen formation. So glycogenesis is also promoted under the influence of this hormone, so that the excess glucose present in the blood will get converted to glycogen. That is a storage form of glucose. Glycogen is a storage form of glucose. As and when the body requires, glycogen can break down and release glucose. So excess glucose, when it is converted to glycogen and it is stored in the body, one region where the glycogen is getting stored is liver cells. So that also will help in reducing the glucose levels in blood. Hope you have no doubts. Any doubts, children? No.
Glycogenolysis, can you explain this one?
Which one?
Glycogen.
Glycogenesis or
No,
glycogenolysis? Okay, glycogenolysis. I'll repeat that. I'll say the difference between both. glycogenolysis and glycogenesis. Glycogenesis means here glucose is getting converted to glycogen. Glycogen formation is taking place, whereas in glycogenolysis, glycogen is getting broken down to form glucose molecules. Okay, lysis means breaking down, genesis means formation. So glycogenesis means formation of glycogen. So it is formed from glucose molecules. Whereas glycogenolysis, lysis means splitting. Glycogen is breaking down. If glycogen breaks down, it will form glucose molecules. Is it clear, Rashmi? Sorry, Nandana?
Yes, ma'am.
Ma'am,
Oh,
could you explain how it acts on hepatocytes and adipocytes?
see, hepatocytes means liver cells. Okay, and adipocytes means liver cells. So what they do is, in which one you are saying, insulin's action or
Insulin.
insulin? So hepatocytes means the cells which make up liver. Adipocytes, adipose tissue is made up of cells known as adipocytes, where fat molecules are stored. So under the influence of this hormone, will promote these cells to use it, take it, absorb it and utilize it. So from the blood, when they take this glucose molecules for utilization, the amount of glucose present in the blood will get reduced. Okay, because in the this insulin hormone acts when the blood sugar level rises than the normal level. So it is more, so too much level, so it should be balanced. If it is too much, more or less, both are dangerous. So when the level increases, so this will try out different methods so that the glucose level can be brought back to the normal. So one method is glycogenesis, means it will promote the formation of glycogen from glucose. That will help in reducing the blood sugar level. Another method is under the influence. So when this hormone acts on adipocytes and hepatocytes, they will start absorbing it and utilizing it. So when they start to absorb it and utilize these, the amount of glucose present in the blood will reduce, and as a result the blood sugar level comes back to the normal. Okay, Gayatri?
Yes, ma'am.
So here picture is also shown. Okay, detailed one you don't have to learn. Okay. They have specified the cells which are taking, fat cells taking in glucose from blood, all that is mentioned over here. So the details you don't have to learn, but what you need to know is that these cells, you can see the picture of both liver cells and fat cells over here. So they will absorb it, and under the influence of these insulin hormone, both adipocytes and the liver cells will absorb glucose from the blood, and that will help in reducing the blood sugar level. Now, when your pancreas are defective, sometimes your pancreas may not produce sufficient amount of insulin, or insulin cannot be properly utilized. There are different reasons, and it will result a prolonged hyperglycemia will result in a disorder known as diabetes mellitus. And here what happens is, through the urine the glucose will be lost, because excess glucose is there in the body, there in the blood, and the glucose starts losing, or urine contains glucose content, and it will result in the formation of harmful compounds like ketone bodies. So that is the main symptom of diabetes mellitus, and to check, you can, using Benedict's test, you can check the presence of sugar in your urine. And earlier people used to add at home also, we were able to do it, some amount of Benedict solution will be added to urine sample, then it will be made or it will be heated, and upon heating it will change the color. The sample taken will change its color, and if it is like some dark color, orange, red and all, it will indicate yellow. So different colors, that a range of colors will be given, and a color chart is also provided along with Benedict's reagent. So you match with the color chart and find out the amount of glucose present in the blood, and if it is more, then insulin injection, insulin therapy has to be followed. Under the guidance or under the directions of a doctor, insulin injections can be taken. And that sometimes by diet control and by taking tablets, you can bring the blood sugar level, or you can keep the blood sugar level under control. But sometimes seeing the amount of glucose in your blood, the doctors might suggest for insulin therapy. And insulin therapy on a regular basis, for some patients they will have to take on a regular basis or on alternate days. So such patients will be trained how to take injection on their own, or they will have to go to the hospital on a daily basis. My grandmother, I still remember, she used to take insulin injection on her own. She will check it using Benedict's reagent, and when the levels are found to be high, accordingly she used to take the injection. That is all under doctor's instructions. So because when it has to be taken on a regular basis, it's not easy to approach a doctor always. But I don't know how the situations are now. I am telling about the case which happened years back, maybe around more than 25 years back. Okay. And now for checking glucose also, we don't have to depend on Benedict's reagent. We have glucometer which you have to poke in your finger or somewhere, then the blood will come, and from that blood automatically the amount of glucose in blood will be read. It is very convenient. Some strips are there which you have to introduce into that device, and it is very easy, very convenient. No need of making taking urine sample, heating it with Benedict's reagent. All that complications are not there now. And it is a disorder due to prolonged hyperglycemia, loss of glucose through urine. That is why the urine is tested, because the urine sample will be having traces or will be having the presence of glucose molecules, and formation of harmful compounds like ketone bodies. Treatment is insulin therapy. So have you understood, children?
Yes,
You
ma'am.
want any clarification? No. You have understood. Now moving on to the next one, gonads, the reproductive organs or the sex organs. There are two types. Testis is known as the male gonad, and ovaries, male gonads. Okay. Two types are there. Testis, IS is singular, ES is plural. Then ovary. Testis and ovary are the two gonads. Two types of gonads found in males and females. First we will see male gonads, testis. Testis as well as ovaries, both of them, just like pancreas, they are having dual function, because they have both exocrine and endocrine function. If you take the case of testis, testis can produce male gametes or sperms. At the same time, sperm ducts are there, so it is having exocrine function, and by means of its exocrine characteristics, they will produce male gametes. At the same time, they can produce male hormones also, like testosterone. So that is why you call testis and ovaries. About ovaries we will discuss later. So testis is also having dual function, both exocrine and endocrine function. A pair of testis is present in the scrotal sac, or you call it a scrotum, and in 10th standard you have learned why it is necessary to have specialized structures known as scrotal sac. And the testis are placed outside the abdominal cavity, because in the scrotal sac the temperature is a few degrees Celsius lower than that of the body temperature. Such a low temperature is essential for the formation of sperms. Testis is made up of seminiferous tubules and interstitial tissues. So, detailed structure of testis, ovaries, etc. you will be learning in class 12. Testis is formed of seminiferous tubules and interstitial or stromal tissues. So, in between the seminiferous tubules, interstitial tissues are present. Then the tissues, the cells present in the interstitial space, or the space in between the seminiferous tubules, you call it as Leydig cells. So, these are present in the intertubular, means in between the seminiferous tubules you can find Leydig cells, and it is these cells that produce male hormones. Male hormones, commonly we call them as androgens, and the main male hormone is testosterone. Okay, once again, gonads in males are known as testes, and these are placed in the scrotal sac outside the abdominal cavity to provide a better temperature for the formation of sperms. And testis consists of seminiferous tubules and interstitial tissues, and in between the seminiferous tubules you can find specialized cells known as Leydig cells, and these Leydig cells produce hormones, a group of male hormones, you call it as androgens. The main one is testosterone. Functions of testosterone. Testosterone will regulate the development, maturation and functions of accessory sex organs. There are, apart from the main sex organs testis, so many accessory sex organs are also there. Epididymis, vas deferens, seminal vesicles, prostate gland, urethra. Epididymis is a structure where the sperms will be stored temporarily. Vas deferens or sperm duct, it is through where the sperms get transferred. And seminal vesicles and prostate gland, I hope you have learned in class 10, together they produce seminal plasma where the sperms are present, which will provide nourishment to the sperms as well as promotes their easy movement. Hope you are remembering. Then urethra, which is a common passage for both sperms as well as urine. So, this hormone or androgens in general, they will regulate the development, maturation and functions of different accessory sex organs like epididymis, then vas deferens, seminal vesicles, prostate gland, urethra, etc. They also play an important role in the formation of sperms or spermatogenesis. Spermatogenesis, genesis means formation. So, they are also involved in sperm formation. It will stimulate the sexual behavior, male sexual behavior or libido. We call that sexual behavior as libido. Growth of muscles, hair, aggressive behavior, that aggressiveness, low pitch voice. So, females are having high pitched voice and males are having low pitched voice. All these are under the influence of this particular hormone, male hormone known as testosterone, or general you can call as androgens. Androgens include a group of hormones, among that testosterone is the main one. Help in anabolism of protein and carbohydrate. Anabolism means building up of protein and carbohydrate. So, once again I will give you the functions. Androgens of testosterone, they regulate the development, maturation and functions of different accessory sex organs like epididymis, vas deferens, prostate gland, seminal vesicles, urethra, etc. And they play a very important role in the formation of sperms. Male sexual behavior, growth of muscles, growth of hair, aggressive behavior, low pitch voice, all these are brought about by male hormones. They also play an important role in the synthesis of proteins and carbohydrates. Okay, children? So, we learned about pancreas and testis. Okay, so two we have learned. So, can anybody tell me how are insulin and glucagon different from each other? The main function. Punya, tell me the main function or the main difference. In terms of function, how do they differ?
Insulin is hypoglycemic factor, which means it will decrease the glucose concentration in the blood. And glucagon is hyperglycemic factor, which helps in increasing the glucose
Okay,
concentration.
okay. Give me any two functions of androgens. Sanya?
They regulate accessory sex organs and
Regulate the development.
development, maturation, and function of accessory sex organs like vas deferens, prostate gland, etc.
Epididymis, etc. Another function, one more function you can say?
They are responsible for the voice of males, hair growth, aggression, and
Okay.
low pitch voice.
Low
Muscle
pitched
growth.
voice, aggressiveness, growth of hair. Okay, okay. Then the last, one more function was there, Gayatri. It helps in or promotes the formation of
Spermatogenesis.
formation of sperms or spermatogenesis. Okay, we'll see next period. Thank you, children.